I've seen you like a million times, but never got to ask you out. What if you don't feel like I do? Would I stand there like a fool? I've never.
Good day, everyone. So in this week's lecture, we are going to continue on the aspect of the gene action. So since we have defined what genes are during the lecture of this chapter, and some differences in according to the different locations of the chromosomes, which is the chromosomes are the, so to speak, we can view chromosome as a kind of an organization or a structure which contains the gene. We obviously all know that the genes are actually the material making the genes are DNA, but in our cell those DNAs do not exist like alone, but they always, especially in eukaryotic cells, those DNAs exist in association with some different proteins, structural proteins. They are mostly those proteins, the names of those proteins, as we have already mentioned, which is the histone. And those histone proteins are actually have some kind of a specific unit of organization we recognize as a nucleosome, and that nucleosomes are some individual unit around which DNAs are wrapped, and they have some kind of the flexibility and also the ability to highly condense their structure, so that this nucleosome, when they have the utmost highly condensed structure, highly packed structure, then now it is the chromosome. But then most of the time, actually, these DNAs plus histone, we recognize as nucleosomes, they exist rather loosely organized. We recognize as chromatin. However, not all chromatins are equal in the degree of a condensation. So some relatively higher in their condensation, then we call them as a hetero chromatin, whereas more loosely organized, just nucleosome structure, then those chromatin we call as a euchromatin. And except for these three locations, the central area, which is a centromere, and the end area we recognize as a telomere, except for these three, the vast majority of our chromosome structure are actually can be interchangeable in its condensation between heterochromatin and euchromatin. And the reason why I am going over this like in review before we getting into this topic of this week's lecture, control over gene expression, is actually this structural aspect of how highly are you organized. I mean, how highly condensed in terms of your the chromatin structure is the key aspect of the so-called control, the ability of controlling the gene expression. This aspect has been neglected actually for a long time. Scientists did not really realize that this condensation and then relaxing kind of by switching back and forth between these two states of chromatin, which all those chromatins are the area containing many genes, and by doing so, those specific the genes within that area have can be influenced in their relative activity of how much how much myself as a gene could be expressed or not. So that's the key feature of this. So having said that, so the overall the flow of information once again is from DNA. Yes, that is the only information, the DNA. And before, actually, the reason why we consider DNA is the utmost importance. Like I compared DNA as a blueprint. The cellular blueprint is because this is the plan. I mean, some area on the DNA, the genes are actually the plan. And according to that plan, the blueprint, and the protein will be made. So that's how the protein coding information is contained in the DNA. That's why we can say this. But this information in the DNA cannot be directly put into the, once again, I like using this expression, the tangible thing as we recognize as a protein. There has to be the intermediate middle guy, which is the RNA. So always the information first handed over to the RNA, and then this RNA is the actually working guy from which actual proteins. So this is the relationship. But then this activity from RNA, still the RNA at the stage of RNA is still conceptual thing, the information. But real physical is something that making differences, the proteins, this conversion, transition of this information into the real tangible thing, which we know as a proteins, happened in this big structure called the ribosome. So with that respect, actually the RNA making is very important. Just because you have DNA doesn't cut anything. So you should be able to convert your information first into the corresponding RNA. Is the relationship is very critical relationship. Cells highly depends on its all activity, which is very important for their survival and maintenance. So this gene expression is, by definition, is the activity of making RNA from DNA template. Here, the reason why it is called the DNA template is something like molding, which signifies RNA is simply just copy, right? RNA is just a copy of DNA, and that area, the particular region that serves as some mold for the copied RNA, well, surely we can call it a DNA template. But here, the thing is, DNA always exists in double strand, a two strands thing, but RNA is a single strand. So here the thing is, so if you are going to make one single-stranded RNA out of double-stranded DNA, obviously we have to consider this is RNA. Then which one, A or B, which strand of the two? So the one, because the relationship between this DNA template and RNA is always, once again, it is a complementary. So it is not exact carbon copy, but rather it's a complementary. That's how, in reality, it works. Just like DNA exists in double-stranded, it is just the reason is because they can have this particular complementary base pair interactions through specific hydrogen bond. The RNA can also be formed. The long strand of RNA can be formed out of a certain region of the DNA, but that relationship is also complementary. So the RNA that is going to be copied, but the information of the DNA from which this particular RNA is made, we call as a DNA template. So then the relationship between DNA template and RNA is complementary. And this, the conversion of DNA into RNA, is called the transcription. Yes. It's like I always try to compare this transcription is in the United States, for example, in college, if you want to request your copy of your whole, like how your performance, your academic performance, that like GPA and all those student records of your performances in all different curriculum courses you took, it's called transcript, right? So you request, you go to the university's registrar's office and then specifically request for, can I get my transcript to send this record to somewhere like for your job application or your graduate school application. That's how you say transcript. And there is a reason why transcript isn't copy your original. And the same thing goes for the case of DNA in gene expression. So you keep your precious original that is acting as your blueprint, the DNA, kept safely in the nucleus, especially in the case of the eukaryote. On the other hand, you only generate the needed copy. So let's say if no, the red, I don't need any red, I need white. So if this is a cell, you have this nucleus in eukaryotes, and you do have all kind of this fuzzy DNA area. So DNA is your information, but then whenever you need only some area, let's say these three regions are the ones containing some necessary information for your gene as genes that to make some specific protein, and those regions are only copied, so in the form of the RNA, but then these copy, which is RNA. But to be more specific, this copy RNA is the messenger RNA that contains such an information. That's why this messenger RNA is called coding RNA, and those copies or get out of. So these are messenger RNA in the cytoplasm. This is the cytoplasm. Then here, each messenger RNA are grabbed by ribosomes, and from each proteins are being built depending on the information, specific information. That's why these are the proteins. That is the reason why these messenger RNAs, since they are exact copy in the form of the complementary base pairs of specific region of the DNA, that's why this messenger RNA is the only RNA that I have introduced 6 different RNAs, including ribosomal RNA and transfer RNA and some other kinds of RNAs. And actually it turns out that they more recently, turns out that these 6 are not the only kinds of RNA. There are some more. So we keep discovering new things. But anyway, that's why only among those, only this messenger RNA is called what? The coding RNA, which means it codes for that particular information needed for making proteins. And once again, the only important things about here is the process of making RNA. In this case, actually, so one might ask, okay, is it like this process of a transcription, so activity of making RNA from DNA, is it only pertains to the messenger RNA only? Answer is no. All kinds of RNA, they are all generated through the same mechanism of this making this complementary copy of that certain area of the DNA. The only difference here is the messenger RNA. As a result, the messenger RNA that generated as this result of the transcription acts as a direct information for making the proteins and all other types of RNAs. The rest of the other RNAs are kind of helping in many different ways, help making this process of gene expression or making protein better and more efficient, or more under the tight control, is how we can say about them. So obviously here this process require some particular enzyme. The name of that enzyme is RNA polymerase, here described as some kind of weird shaped ball, like I don't know how to call this. In reality, of course, that shape of RNA polymerase is much more complex and difficult to imagine because it is highly complex structures. But simply to simplify the structure here, all we need is hey, there is some kind of specific enzyme catalyzing this procedure. Every activity, every metabolic activity in the cell requires this involvement of various type of enzymes, as we know. Now, the second the important information is, okay, so there are many genes scattered around on the DNA, on your chromosome, but then when they are going to be transcribed, okay, when those RNAs are being made, do they do this like in terms of a synchronized and all together and then stop? No, that is not the case. So some genes are active in the certain particular timing, while some others are not active. And also those differences can also be seen in different types of tissues in the multicellular organisms like us. So what we can say about this activity, the transcription, is not all genes are active, okay, in a given time or in a given location of your body. So simply put, not all genes are active in any kind of observation point in some certain specific genome, right? So there is some distinction between active genes and inactive genes. So what are the active genes? Those genes that are actively transcribing, that making RNAs, many copies of RNA. Usually when you make copy, you don't really simply generate a single copy. Okay. Several, at least several hundred, usually, copies. Even beyond several thousand copies could be made in the form of messenger RNA or in the form of some other different type of RNA. So, on the other hand, inactive, or sometimes it's called the silent genes, are when certain region there is a gene, definitely that area is taken up by the gene, but it is inactive. It is just simply not doing. It is maybe it's like on vacation. It's doing nothing. No copies of RNA, or sometimes virtually means no copies at all or very little, right? So, but then what does determine the difference between active and inactive genes? Am I destined to become an inactive genes at the birth at birth? No, it's not. In a given gene, actually, it may be active depending on different timing or depending on different location, as I said. So it all under the specific planning and then different specific control. Here comes the very important concept of differential gene expressions. So this differential gene expression means by which cell, usually this differential gene expression can be more obviously can be observed in the multicellular organisms. However, just because you are a single cell organism like bacteria or yeast or protist doesn't mean that you don't have any this kind of a differential gene expression. Even as a single celled organism, you can have a single celled organism at least can have this feature of timing. You let the certain group of genes expressed in a given specific timing, and then later you shut them down and then activate other set of genes that are their very usual casual way of the control of gene expression. And the difference, the only difference actually in additional difference in the multicellular organism is that on top of that, this multicellular organism, because they are the different tissues type, then this differential gene expression can also be observed in different types of location of the cell types. Okay, so this differential gene expression usually you can see very clearly in the multicellular organism, and also the key feature of the multicellularity. What makes this multicellularity as a multicellular organism? What is very essential things? It's the ability of this differentiation, which is absent, entirely absent in any single-celled organisms. Yeah. So, in other words, this so-called differential gene expression is the one that make the multicellular organism possible. If you consider that just because you are kind of individual consisting of so many cells doesn't mean that you are a true multicellular organism. You are nothing but, if that was the case, you are nothing but a cancer. So you have to demonstrate this differentiation, which means certain different area location shows differences in shapes, appearances, and also the functions. That is called the differentiation. But then how do you achieve this differentiation? This differential gene expression is behind this so-called differentiation. And so then, this differential gene expression, which is simply turning on certain sets of the genes in a given specific location, while some other location, that particular same set of genes are not active, is a very good example and very straightforward example of this differential gene expressions. But then that is not the only example of a differential gene expression, because you can exercise such differential gene expression in different timing. But then these timing differences in gene expression can also be observed in this single-celled organism, is my point. Okay. So then, before we just jump into this, let me reiterate the very clear example of, I think I've mentioned at least once before about this. So what is the best example? I shouldn't say it is the best example, but one very clear example of such differential gene expression. Think of this hemoglobin. Is the protein catalyzing the transfer, carrying the oxygen. You breathe in, then the oxygen, we pick up the oxygen. Why do we need an oxygen? Because in every different cells in our body needs that oxygen for our cellular respiration, the aerobic respiration occurring in the mitochondria, right? So who is going to deliver that oxygen to you as a mitochondria? That is the hemoglobin, right? The protein hemoglobin, however, so do we have this? Does every cell in our body has this hemoglobin gene? Yes, every cell in our body has this hemoglobin gene. However, only the red blood cell. All other types of cells and tissues, even though they have this hemoglobin gene, the gene making this hemoglobin protein, but that gene is not active permanently, so to speak. It's kept silent. Only active gene, only type of cell within which this hemoglobin gene is active is the red blood cell, right? Then how do you control this? How do you make this, well, certain area you do still have the gene, then they are not active, and some other type of tissues, you have the genes and then they are active. And that is the actually key feature of those differentiation. And let's say you have 100 genes altogether. Your total 100 genes at birth, like your zygote, your fertilized cell, from which you keep dividing. So cell division will produce so many different cells, making, right? So certain area will develop a specific tissue A. Some other area will develop tissue B, etc. So that's the basis of this so-called being a multicellular organism, like tissue A and B. And then both tissue A and tissue B inherit same 100 genes originating from the first cell, the zygote. Okay. However, the way you exercise this differentiation is, for example, in tissue B, you have one specific 10 out of 100. So this particular 10 is only active. Okay. On the other hand, in tissue B, another different 10 gene out of the same 100 gene is active. Obviously, then the gene product, the different types of protein will be made between tissue B and tissue A, and that is the basis of making those two different types of tissue different, and that is called differentiation. And the mechanism enabling this differentiation is differential gene expression. Okay. Then the question is, how do you achieve this the differences in differential gene expression? It has to be something that's through differently controlling the gene activity, while you are shutting down the genes in certain area of the tissue. On the other area, you are making those exactly same type of genes highly active. To understand that, the first, like, what's there? What are those factors that are needed for this gene expression? That's what I'm going to explain right now. So first, it's so obvious that in order to start this transcription, which is the RNA making process, you do need the main guy, the main character, the guy which is needed for this catalytic activity. The name is the RNA polymerase. So this RNA polymerase is the enzyme catalyzing this activity of the transcriptions, right? However, so then, can RNA polymerase, so like simply put, if you have 100 genes, and this RNA polymerase can transcribe all these different 100 genes, there is actually some deviation. So, like in prokaryotic cells, which are simpler than the eukaryotic cells, that is the case. So the RNA polymerase, basically you do have one single type of RNA polymerase making the RNAs of all different kinds of so many different genes. Right, sometimes you have a little bit of a different adapter modules in the case of prokaryote, but basically the core unit of that RNA polymerase is the one identical type. However, in eukaryotic cell, which have decided to become a little more complex, there are 3 different types of RNA polymerases acting on. Like they have their they divided their roles. Like, okay, I'm as a RNA polymerase 1, then I'm going to make only ribosomal RNA transcription, for example. Hey, I'm RNA polymerase 2, number 2. I'm going to make I'm going to be responsible for the RNA production of only the messenger RNA genes, cell RNA. So there are 3 different types of RNA polymerase exist in eukaryotic cell. But having said that, basically, how many different genes are there in, for example, like how many different protein coding genes in our human cells, like tens of thousands, right? So then all those tens of thousands of different genes, protein coding genes, that single one kind of RNA polymerase will take care of their transcriptions. So then let's consider this. Here are the reasonable chromosomes. So this is a chromosome. DNA, actually, the DNA. But not all area of your chromosomal DNA are occupied by the genes. So what's the difference between gene and the non-gene area, the intergenic region? So here, certain area like this area is a gene A. Another area maybe gene B. Also other area gene C. But actually the majority of the area are not occupied by the gene. And so, for example, this area, why not? Why not me? How can I become a gene? What is the requirement for like being a gene? If this area asks, then what would be the answer? So that up until now one thing is clear, that all these genes will make the RNA, right? So we already defined the broad definition of the genes. The area gene, the area that can potentially make RNA. Like, in other words, the area that can do the transcription. So there's still questions remains. Why not me? Why can I make the RNA? So the answer is, okay, what it takes is, so in order for you to make the RNA, you have to be able to be friends with the RNA polymerase, right? This RNA polymerase, the enzyme, somehow will act on these 3 areas, but for some reason here it cannot. So what's the difference? Why doesn't this RNA polymerase visit me? Could be a complaint. maybe a valid complaint of this area? Here, the answer is this. The keyword is this: promoter. To start transcription, the RNA polymerase needs some specific element. RNA polymerase has to bind to a specific area of the gene. So let me give you a ugly graphical illustration of what this promoter is. So let's say this area is once again red. So this is the area of DNA. And if this is the gene, so which will generate exact copy of RNA, right? Gene is DNA. And so that means RNA polymerase. I will abbreviate this RNA polymerase as RP. RNA polymerase has to come and do this activity of copying the template area of the gene, DNA double helix. Then to do so, RNA polymerase has to be able to bind, to sit on. So it's like if you are an RNA polymerase, you need some kind of landing spot. RNA polymerase is floating around on your cell in 3D. Try to visualize a 3D way. Then RNA polymerase has to recognize the certain area and then has to be able to land on it and then start. Actually, the starting procedure takes this unwinding of existing this DNA double helix. Then excuse me, and then can I open up this so that RNA polymerase can choose one of these two strands and then keep inserting the RNA nucleotide according to the specific DNA sequences, which make this temporary DNA and RNA the complementary relationship. So anyway, this area, which is outside of this area of when we say this is a coding region, which defines the start and the end of a certain gene, which will exactly correspond the length of this RNA. But then RNA polymerase, if this is RNA polymerase, has to sit on the front line of this starting point of these transcriptions. Then this area of the DNA to which this RNA polymerase sit, which means bind, we call this as promoter. So in other words, the promoter is a DNA, specific DNA sequence that is being recognized and bound by this enzyme RNA polymerase protein. So if you have this, then RNA polymerase can sit on it and then start working on this transcription. Unfortunately, if you don't have this the combination of DNA sequences, then you are tough luck. Then RNA polymerase will never visit you. So that is something that makes the difference between gene and non-gene. Yeah. So that is the definition of promoter, and what we can say is promoter is located at the upstream here. What do I mean by the upstream? If this is the coding area, so which is the starting point of the gene, RNA will be made. Okay, we usually describe as this. And this area, so this is upstream. If you consider this RNA as some kind of orientation, the start of the RNA is the 5' end, and the end of the RNA is also 3' end, according to this, the designation of this, the nucleic acid orientation. You know what I mean by this 5'. So 5' phosphate and 3' hydroxyl group is so like DNA is organized, DNA double helix organized into this 5' to 3' end and 5' to 3' end in this anti-parallel ways. And RNA also has this kind of, if the RNA is going to make this the complementary base pair relationship with the area of a single-stranded DNA, they also have to be in this anti-parallel way. So one is going from 5' to 3' direction, and the other one is going reverse from the other end of the 5' to 3' end. So that's the only way any nucleotide, any nucleic acid strand can have this particular complementary base pairing relationship. So if this 5' end of the RNA start and the end of this RNA is 3' end, if we describe this RNA in this way, then this DNA area, the gene area, the coding area, where is the location of this promoter? Then you can say usually it's an upstream of the coding region, which means 5' end upstream. Okay, so this is a up before the start. And then what do you call this, the other area beyond the end of the coding area of the gene? Then downstream. So that's the way of designating the relative location around the certain coding area of the gene, upstream and downstream. Yeah. So once again, the location of the promoter is always right upstream, 5' upstream of the coding region, right? Here, this diagram, this figure shows a typical eukaryotic gene organization structure, which is quite different from a typical prokaryotic gene organization scheme. So what I have already said that promoter area is not included in the coding region of. So here, this marks the coding region of the gene, and so the product of the gene, this particular gene, would be equal in length, equal to the coding region. So the RNA, this RNA length initially, at least initially, will be identical to that of the coding region of the gene. So that's the definition of the coding region. Okay. Here, what I'm going to show and explain is the difference between coding region versus non-coding region of a gene. Okay, so here non-coding region, I'm not saying this non-coding region as a known gene. The purpose of this explanation is try to define actual the one unit of the genes, like what is included in a gene. Is it only some coding area of the genes, then that's it, or do we need something else? Obviously we have already witnessed that in order for any area of the DNA to become a gene, you need a very important element, DNA sequence element, which is the promoter. However, the promoter is not a part of the coding area, which means if you look into this RNA sequence to find where is the promoter sequence, then you can't find it because it is not there. So promoter sequence is not included in the product of the gene. So which means promoter is not a part of this coding region, shows that you do need something extra, like additional area for a gene. So the coding area alone is not something that constitutes a gene, is the point. So although the promoter is not a part of the transcribed RNA, that you cannot see the promoter in the product RNA, but still any gene cannot function. A gene cannot be expressed without its promoter. Okay, so that's the first thing that we have to understand. It's a long procedure to reach the whole entire picture. It's not an easy concept, but without this, we cannot fully understand what is actually basically in essence behind the control of the gene expression. So we only just took just a baby single first step. There's something else actually. The RNA polymerase is absolutely required things. Every gene needs RNA polymerase as a starting the main enzyme. However, if you only have this RNA polymerase alone cannot do anything. You do need some helper. So that's the dilemma of RNA polymerase. So you need some other proteins other than RNA polymerase. Those are collectively called the transcription factors. Okay, there are many different types of proteins function as the transcription factors. So here, the contrasting thing is, although there is only one single kind of RNA polymerase, but many different types of transcription factors, and depending on who you are as a gene, different genes require different kinds of at least one transcription factor for a proper function. Once again, RNA polymerase alone will have a very sluggish, poor job. So virtually you cannot see any gene product without any transcription factor proteins that act as a helper for this action of RNA polymerase. Why? Because RNA polymerase is very unstable. The transcription is a very risky business. A lot of times the RNA polymerase, while they are trying to copy a certain area of the DNA by inserting some RNA nucleotide, it can fall from. It's like a rope riding. It can be viewed as some kind of rope riding, acrobatic activity. So you can easily fall downhill. Then, oh shoot, then you have to climb again and then do it again all the way from scratch. That will severely slow down the completion of transcription. But if you have a lot of different protein factors called the transcription factors, it can stabilize such things, so it can go all the way down to till the end of the gene much faster. That means certain area of the gene RNA polymerase will recycle several times to generate many copies of these things. That's how you generate multiple copies of RNA out of a single area of the DNA. So that's why transcription factors are needed. That's the reason A. There is another reason. There is another very important reason why transcription factors are also needed for this gene activity. So collectively, whether it is a reason A or B, what you can say is collectively the reason why they are needed is the efficiency of the gene expression. The presence of the transcription factor, they are proteins, greatly enhance the gene expression efficiencies. One reason A is stabilizing the RNA polymerase to bind the promoter area. It can hold tight. The reason B, the second reason why transcription factors are so essential is it's just about the matter of accessibility. I have shown already once this feature in when I was showing the difference between heterochromatin and euchromatin. Let's say certain areas are, right now is an heterochromatin. Heterochromatin, so that means it's covered. This is the gene area, but unfortunately this area is all covered by so compact. These are all nucleosome. Basically, nucleosomes are histone proteins. So even though inside of which this is the area of the gene, this is a gene. However, this gene area, RNA polymerase, if you have this RNA polymerase over there, RP, try to get access to this particular area, which is the promoter. So RNA polymerase, this RNA polymerase has to find and then land on it, but this promoter is covered with the nucleosome. So it's inaccessible now. And RNA polymerase itself alone cannot clear this nucleosome and get over, it can move over, get away from, and then clear the way, and then it can sit on to this promoter area. RNA polymerase alone cannot do this, unfortunately. But transcription factors can. So if you somehow can bring your friends transcription factors, several different kind of transcription factors, TF, then it can clear. This transcription factors can clear the area. Wow, for some reason. And then this nucleosome will be absent now, thanks to this transcription factors activity. Now showing so-called naked naked DNA with the promoter area is now exposed. So then RNA polymerase now can sit on here and then start transcription. The second reason might be more important for the necessity of the transcription factors, and also so whether a single transcription factor protein can both of this clearing and then stabilizing of these two functions. Not usually. That's why you do need several different types of transcription factors are needed. So certain group of transcription factors doing its job of clearing this area, while some other transcription factors are stabilizing the RNA polymerase. But that doesn't mean that a single transcription factor can do both of these things. But overall, those little difference details informations are not necessary for you at this level. So simply put, once again, clear the way. Once again, to summarize, gene expression requires transcription factor proteins in addition to the RNA polymerase, because RNA polymerase alone, the speed or efficiency of transcription will be very poor or low for many reasons. But in the presence of a transcription factor protein, it can greatly enhance such activity by two reasons. One is stabilizing the structure of RNA polymerase. Number two is if that area is covered with the nucleosome, which means that heterochromatin, then those transcription factors can convert this heterochromatin into euchromatin. And that is the reason why, actually, for any gene activity, first off, to become a euchromatin is very essential. If you are right now currently in a state of a heterochromatin, like this heterochromatin, then any genes included in this type of status, then genes are not active. And that is the reason why that the constitutive heterochromatin area, like the centromere and two telomere areas, you cannot really find any genes, virtually no genes. Although some centromere area you may be able to find a few genes, but basically these areas are almost permanently exist in as heterochromatin. So there is no way, actually, if you ever actually put any meaningful gene over there, their activity will be always permanently be shut off. That's why. And so this is how like if your DNA is in this heterochromatin, tightly packed, compact area, then transcription factor, with the help of some other enzyme, it can convert this area, like lacking any nucleosome. So you can see this area is naked now, revealing this promoter, and now that's the beginning of gene expression. So because of that, what you can say is, therefore, the activity of a gene is absolutely dependent upon the transcription factor. And these transcription factors also have to work together with the RNA polymerase on the promoter area of the gene. So, what having learned that, what we can say about this active gene and inactive gene is, if a gene is active, that means transcription factor is now bound to the promoter, not promote. Transcription factor is bound to the promoter together with the RNA polymerase. If for somehow, if a gene still, even if it's the same gene, but if that gene is inactive, which means not making any RNA copy, that means even though there might be a RNA polymerase available over there, but still transcription factor is not there. So that is the difference. And whether certain specific transcription factor can bind to the promoter area or not is highly dependent upon the DNA sequence of the promoter area. So in summary, let's say this is the coding region of the DNA, a gene. So this is coding region, say, of a gene. So through transcription, will generate the RNA, which is equal in size. So the transcribed RNA will exactly match the size. So this is messenger RNA. Let's say this is a messenger RNA. Then for this, you need some element which is necessary for this transcription to begin, which is a promoter, right? So to speak. So this is so what is a promoter? A binding site. So let's just binding site for the RNA polymerase. And this RNA polymerase will catalyze this transcription. However, the point is you do need some extra to become, for this gene to become actually the really active gene, you need something else, which are the transcription factors. Then the question is, where do these transcription factors come in together with the RNA polymerase? No, it is not. Usually it is also some other DNA sequences outside of this coding region of the gene. There are some specific sites. Let's put it this, okay. Some of the specific area where these transcription factor, one such a transcription factor, another such transcription factor. So all these transcription factors, they are proteins. are required. Why? Because these transcription factors can help in either stabilization of the RNA polymerase or clearing the area so that RNA polymerase can have a clear path without having any blocking nucleosome for this action. So then what do you call, although it is not really such a necessary thing for at this level, what are these yellow area or yellow element? If this white area, this area was called the promoter, which is serving as a binding site for the RNA polymerase, then those DNA elements serving as the binding site for the transcription factors, this yellow area, are called enhancers. So it's just a terminology. So the promoter and enhancers are both the specific DNA sequence that are required for these proteins, including the RNA polymerase and the transcription factors, to bind on specifically. So in other words, if you don't have any DNA sequence that can serve as a promoter, then no transcription whatsoever can occur because RNA polymerase cannot bind. So that's why some area of the DNA cannot function as a gene. On the other hand, if you do not have any enhancer sequences, usually located further upstream of the promoter area, then you cannot actually have a efficient transcription. So that area are going to be kind of always inactive as a gene. And third, even if you do have both promoter and enhancer sequences on for the genes, by the way, mind you, those enhancer and the promoter DNA elements are not going to be found in the product of the transcription with the RNA once again. So if the coding area, RNA, then here you cannot find any promoter sequence or enhancer sequences because they exist outside of this is a promoter and this is enhancer, and maybe this is an enhancer. Usually enhancers exist in for the upstream of this coding area. So this is a 3' end and this is a 5' end of the gene. That's how you describe or designate the structure of a gene in eukaryotes. And also the prokaryote case, the same thing goes true, but in prokaryotes they don't have exact enhancer sequences which is 100% equivalent to the eukaryotes. So let's forget for a moment about the case of a prokaryote. Let's just focus on the case of eukaryotic cells gene structure. So these are those DNA area that are not found in the gene product, which is the RNA, but still necessary for any gene to function. So these type of area, including the promoter and enhancers, they are collectively called the control element or regulatory element. They are kind of supporting actors, supporting staff in play or in movies. On your interface over like a movie screen or anything like that, what you see is the actors, main characters and all actors. But to make those movie that being made by those actors possible, so then there are so many other supporting steps working hard, and behind the screen is probably kind of similar analogy. So if you are going to define a certain area of DNA as a gene, you should not exclude. You have to also include these control or regulatory elements, because these regulatory elements are the ones that are needed for inviting or recruiting, anyway, whichever you want to prefer to call, recruiting those the protein factors, including the RNA polymerase and this transcription factors. And sometimes actually these regulatory elements, the area of these regulatory elements is much larger or longer than the actual coding region of the gene is was been found in the case of most of the eukaryotic genes. And these promoter and enhancer sequences are like that specific, those elements, DNA sequence elements, are highly sequence specific. In other words, for different genes, the promoter sequences are kind of universal. That means single RNA polymerase act on all those promoter sequences present in every different genes, so to speak. Okay, although like I said, there are 3 different kinds of RNA polymerase that exist in the case of eukaryotes. But overall, like if you think of all those messenger RNA coding genes, then the thing is one type of RNA polymerase is working on all different messenger RNA genes because they share same promoter sequence. However, in the case of enhancers, they are variable for different genes, so they are highly specific. For a given gene, which means, what does it tell you? That means for different genes, the transcription factors that working on different genes are different. Different gene utilize different kinds of transcription factors. Of course, some group of genes may share certain type of transcription factors. It's too much details, but like just in case you were kind of confused, so what's going on? I mean, transcription factors are they all just like RNA polymerase, so that they can be used in all other different genes? That is not the case, and that is actually the secret, a trick through which this so-called differential gene expression can be made. That's why I touched this area, this information. So the specificity of the transcription factors determines the gene expression, unique gene expression profile, so to speak. So in other words, anyway, so there are some these sequence-specific elements like promoters and enhancers is probably the only messages that you can carry on out of this. So sequence to which transcription factors binds is called enhancers. And if there was an angel, then there was a devil. So some DNA elements also recruit some protein factors that are acting against this transcription activity. Okay, so what you can say is some proteins inhibit transcription of a gene, just the opposite to the function of the transcription factors, and those proteins are called the repressors. So like there are two opposite forces. One is transcription factors, and the opposite to this is repressor. And both kinds of these are proteins. One is helping, the other one is blocking. And so for those repressors, they also require some sequence-specific binding site. So this sequence-specific binding site is called silencer. So it's always so. What is some kind of a common rule that you can find over here? Instead of digging into little trivia details, which are largely unnecessary for anybody, so the kind of rule that we can find over here after all this is that all this interaction regarding this gene regulation, gene transcription and regulation, is the result of always the association or the interaction between some specific part of the DNA acting as a gene and some specific proteins. So here comes always the relationship between protein and the DNA interaction is what makes all this necessary control in what to make, how to make, how much, what to make, how much you make in terms of this gene product is probably the take-home message of all this. So if we so this is once again as a kind of summary figure. So here is the coding area, which will exactly correspond to the gene product, RNA, and in addition to that, what you have is this other area, the regulatory area here, usually present in upstream of any gene, and the other side we call it a downstream, like that. So like single gene unit is defined as the coding region plus regulatory element is what all together having. So if you want to call a certain area of your DNA as genes, then you have to include a coding region plus a regulatory element, like that. So this coding region is also another things that I'm going to touch was the coding region itself. If you look into further closely, there's several different area divided into several different area. Like one is exon, and the other one is intron, and exon, and intron, exon. What are these? So as a result of the transcription, what you get is this, the first product, as an RNA. Here, this is the case of eukaryotic messenger RNA. Only, like even though the genes, the eukaryotic genes, but the case of ribosomal RNA genes or the tRNA genes don't have this type of blocks called exons and introns that we are going to take a look at this. What are these? But anyway, this is the case of a messenger RNA in eukaryote, and the messenger RNAs of prokaryote, bacteria, do not have it. Such things like exon and intron and exon introns. What are these? This coding region consists of what? Just follow me. Exons and introns. Yeah, it looks like. Starting from this is a starting point of 5' end, and this is the end of the gene's 3' end, and then certain area of the coding area is defined as an exon, so-called exon. What is that? And that exon is followed immediately by introns, and then goes on then another exon and another intron and an exon. Although all this DNA sequence is kind of random or unique, but kind of arbitrarily. No, it's not arbitrary. Certain area would definitely define that area of the gene as an exon, and the other one is an intron. We will get to that. What's the difference between exon and introns shortly after finishing this. And speaking of a control element, the regulatory element of the genes, the promoter is the binding site for the RNA polymerase. But also, there are some other regulatory elements in upstream regulatory elements that enhances the binding site for regulatory proteins, the transcription factors. So this regulatory region of a gene that contains the control element, their function is recruiting the various regulatory proteins, including this. Some transcription factors acting very closely in close proximity with this RNA polymerase. This is RNA polymerase, while some other transcription factors working further upstream of this transcription site starting point. That's what it's like. Okay, so that's for the case of a regulatory region for mainly recurring the proteins. What about these exons and introns here? Now, to understand the difference between exons and introns, take a look at this. This is the immediate gene product of eukaryotic messenger RNA, which several times I said the length of the initial gene products of eukaryotic messenger RNA is, of course, I mean, that's the same thing. That goes true for every other different type of genes in both in prokaryotes and eukaryote. The size or length of the gene product is equal to the coding area of the gene, obviously, right? But in the case of eukaryotic messenger RNA, wait a minute for a second. The initial product of this transcriptions goes through a further process of editing. It's collectively called the RNA processing. Take a look at this. If you look at this, this is called the primary RNA transcripts in the case of eukaryotic messenger RNA. But this is the final mature messenger RNA. Final mature RNA, that is actually the one that you see in the cytoplasm. Cytoplasm, what happens is the ribosome will take charge on this final messenger RNA product to start the conversion process of this information into the product of a protein. So that's something happened in the nucleus. Then what you see is some kind of further processing. Here comes, if you distinguish the certain different area of your primary, the first RNA product, as the red block area as an exon, and this pinkish area is an intron, and another red area in exons, then what you have left as a final product is all this the intron, the area representing the intron area, are all removed. Introns are all removed, and the remaining exons were put together. So that is called the RNA, the exon splicing, putting together to make this final finished product of a messenger RNA. So the one thing is, if you compare the length of a final mature messenger RNA with that of the initial coding area of the gene, like that, then is obviously there is a big gap. This final mature messenger RNA product is much shorter than the original, the coding region of the DNA, because the intron areas were all removed. So without getting into further things, at that point we can distinguish the difference between introns and exons in eukaryotic messenger RNA, as like intron is, what you can say about intron is a segment of the messenger RNA coding region that is absent in the final mature messenger RNA product. So those introns are removed, are not there. Whereas the exons, then, how do you define the exons? The exons are segment of the messenger RNA, the coding region, that still there, that remains in mature messenger RNA. So that's simply the most straightforward way of defining exons and introns. I took so much time in explaining the nature and organization structure of the gene organization, especially in eukaryotic genes. So next, before we get into why we have, especially in the eukaryotic messenger RNA genes, why do we have these seemingly unnecessary elements like introns? But before that, we are getting into, let's take a look at first how the basic principle of this, the differential gene expression, which is very important, critical for maintaining the multicellular organisms, the differentiation. But I took so much time in, like I said here, in explaining this, so probably I will have to stop here for the first session of the lecture, and then I have to continue this in the following second session of the lecture. Okay.